Hello everyone and welcome to today's lecture. In this session, we'll be learning about IPTG induction, a widely used method for expressing recombinant proteins in microbiological experiments. Today's lecture will cover three key parts. First, we'll introduce what IPTG is and its role in protein expression. Next, we'll look at how IPTG functions at the molecular level through the log occurrence system. Finally, we'll understand how this process fits into the broader context of microbial experiments. Let's begin with a quick overview. IPTG is a synthetic compound used to trigger gene expression, especially when producing recombinant proteins. Recombinant proteins production is when we insert a target gene into the host cell, like E. coli, so that it produces a specific proteins for us to study or use. IPTG is most commonly used in E. coli expression systems. In these systems, plasmids are used to carry a target gene, and gene expression is regulated by a system called the log operand. So, what is the log operand? It's a group of genes in E. coli that help the bacteria digest lactose. However, if lactose isn't present, the cell saves energy by turning those genes off. This on-off switch is controlled by a repressor protein and is the key to how IPTG works. Now, let's take a closer look at how IPTG activates gene expression. IPTG is a chemical analog of allolactose, which means it mimics the natural inducer in the log operand system. However, IPTG has one major advantage that it is not broken down by the bacterial enzymes, so it stays in the system and keeps the gene expression turned on continuously. Here's how the process works. Normally, the log repressor protein is bound to the operator system on the DNA. This physically blocks RNA polymerase from transcribing the gene. When IPTG is added, it binds to the log repressor. This changes the shape of the repressor protein so that it can no longer bind to the operator. With the repressor out of the way, RNA polymerase can now bind to the promoter region and start transcribing the gene. That leads to the production of mRNA, which is then translated into our target protein. As a result, the target gene, often a recombinant protein, is expressed in large amount. IPTG induction allows precise control of protein expression in a recombinant system. It's a key technique widely used in molecular biology and biotechnology. Thank you for your attention.